So there are many reasons why this microbiome project was beneficial. It allowed us to look at the types and proportions of microorganisms living in a given location, such as the stool samples of specific organisms. Analyzing the microbiome of an animal allows the scientist or person doing the research to see the diet, environment, and several aspects of the animal's health. To see how these organisms are influenced by their health allows us to see if the food they are eating within their environment is beneficial to their survival. The question our group raised was, how does the age of an organism affect the types and amount of microorganisms within the host? Our hypothesis was the amount and strength of microorganisms in the host will increase as the host size increases. The 4 animals we tested for this project were 2 dogs, a pig, and a hedgehog. The reason we chose these animals were due to the fact that all are mammals, but their environments are not the same. The results that we obtained from doing this experiment were not seen through the animals we tested, but instead we analyzed information of 2 different dogs that have already been tested. One was a flat-coated retriever and the other was a husky. The number of reads for the flat-coated retriever was 18,028, but the number of successful reads from the total number of reads was only 17,852. And the total number of reads for the husky was 3,023, but the number of successful reads was 1,850. The significance of these reads were to show that the amount of bacteria within the gut after sequencing. After using this data, our hypothesis was supported because the number of successful reads was greater in the larger host, the flat-coated retriever, than in the husky. So this experiment was to test the gut microbiome of our samples, and there is a difference between a gut microbiome and just a microbiome. So a microbiome is less specific compared to the gut microbiome. A microbiome is the microorganisms in a particular environment, but specifically, a gut microbiome is the microbiome population living in our stomach and intestines, which contains tens of trillions of microorganisms, including at least 1,000 bacteria present. The reason why these bacteria within the gut are so important to maintaining a healthy organism is because an imbalanced or ineffective microbiome can cause diseases such as gastric cancer, obesity, irritable bowel syndrome, or several other diseases. Different size hosts will come with different types of bacteria. So for our project, our question was, how does the size of an organism affect the types and amount of microorganisms within the host? And our hypothesis was the amount and strength of microorganisms in the host increase as the host size increases. So for material and methods, the 2 animals compared in this experiment were a 4.5-year-old flat-coated retriever and a 3-month-old husky. They are both female and neutered, both in very good health. Their diet may differ a little. The retriever was fed a diet of yogurt and dry dog food, while the husky was fed milk bones, rawhides, and grain-free dry food. Their environments are slightly different. One is both indoor and outdoor, and the other is just an indoor pet. The 3-month-old husky also has coccidia, which is an intestinal tract infection that may cause diarrhea. They had a weight difference also, but this is mostly due to the age difference between the 2 dogs. So during our first week, we were testing the 4 samples that we originally were using for this experiment, which was the pig, the 2 dogs, and the hedgehog. So in week 1 of our experiment, we isolated the DNA from fecal matter, and we used the Quiagen kit in order to do this. And then we concentrated the DNA tenfold using the Zymo kit. That way we could concentrate the DNA 10 times more than originally. During our second week, we used absorption spectrophotometry to determine the concentration of our DNA and to see if there were any contaminating proteins or RNA. And we used this using the NanoDrop 2000. And because DNA can absorb light at 260 nanometers, we could use that to determine the concentration and to determine the purity of the DNA. And to do this, we use the ratio of absorbance of 260 nanometers to 280 nanometers, since proteins absorb light at 280 nanometers. The normal range of pure DNA is at a ratio of 260 to 280 is 1.5 to 2.2. And so after we tested the concentration of our DNA, our next step was to amplify the 16S ribosomal RNA gene using the polymerase chain reaction, or the PCR. And to start the amplification, the target sequence is denatured, and then by raising it to 95 degrees Celsius, then brought down to 54 degrees Celsius to allow the primers to bind. And then once they bind, the temperature is raised to 72 in order for DNA polymerase to bind and start synthesizing. And we do this cycle 25 times, which will make billions of copies of the sequence that we were looking at. During week 3, we used the gel electrophoresis to determine whether or not the products of our PCR reaction worked. And this was done using an agarose gel, and this allows the DNA fragments to move when we run an electrical current of 250 volts through it. And to remove the unused primers and other reagents, we used the Agincore Ampere XP kit to clean up the reaction. So this was the PCR cleanup phase. And we were able to identify what sequences in order to identify what sequences came from which animal, we added specific tags to the end of our samples using indexing PCR. And after preparing the samples, they were put in a thermal cycler for 3 minutes at 95 degrees Celsius. Then for 8 cycles, there is an added 30 seconds at 95 degrees Celsius, 30 seconds at 55 degrees Celsius, and 30 seconds at 72. And it continues at this for 5 minutes and then holds at 4 degrees Celsius. So during week 4, we performed the PCR cleanup again. In order to determine the concentration of our amplified samples, we used a fluorometric assay with the Qubit fluorometer. And we use this because it is more sensitive and accurate than UV spectrophotometry, and it can distinguish the difference between DNA, RNA, and proteins. And then after this, our samples were sent to be sequenced using the Illumina MiSeq NextGen DNA Analyzer. So for our results for week 1 with the NanoDrop, we have our animals listed, their sample number, and then we had the concentrations of our starting samples, and this table also shows the 260 to 280 ratio. So you can see here that all of our samples were considered pure because they were in the 1.5 to 2.2 range, and our lowest actually was 1.7, so that was really good, and our highest was 1.97. So we were able to continue with our process of doing this. And so next you can see we have the images of our of our gel electrophoresis, and you can see that each band here signifies a different sample. So we have our positive, our negative, the hedgehog, pig, and both of our dogs. And you can see that the only one with an absence of a band is the negative control, which is a good thing, because if we had a band here, that means that that the negative control did contain DNA, which means something would have been cross-contaminated. So it's a good sign that we didn't have anything in the negative control. But for the positive control, we have a band because we used DNA from E. coli, and the rest of our samples should have bands along the 550 base pairs because they did in fact contain the DNA of the animals that we were testing. And all of our samples lined up with the 550 base pair mark, which is exactly what we were looking for, so that was good to see. And so the next table shows our Amplicon PCR reactions, and it shows the quantifications of the cleaned up Amplicons. So here we have the microliters listed according to our animals and their sample numbers. And so our next table down here shows the quantification that we use for our indexing PCR reaction. And since we had no errors in our samples from previous steps, we were able to use all of our samples for the indexing PCR. So for our fluorometric quantitation, we have here the animals, their sample numbers. So for the hedgehog, we got 48 nanograms per microliters. For the pig, we got 18.4 nanograms per microliters. For the dog, we got 3.54 nanograms per microliters, and for the second dog, we got 40.8 nanograms per microliters. And now we'll be analyzing the results from a previous study because our samples were unable to be sequenced. So here are the results from the animals that we did not test, so the flat-coated retriever and the husky. So when we put these through the sequencer, for the flat-coated retriever, we got 8,028 total reads, with 17,852 being successful. And for the husky, we got 3,023 total reads with 1,850 being successful. And so for the results for our flat-coated retriever at the kingdom level, Oh no, the lights turned off. Sorry about that, Dr. Warren. Okay, so for the kingdom, results for our flat-coated retriever. Let's see, we have them down here. Okay, so you can see that bacteria make up 99.02% of what was able to be classified. 92 or 99.02%. Unclassified at the kingdom level is 0.98%, so very little was unclassified at the kingdom level. And we found that viruses made up 0.01%. So in the number of reads, we had exactly one virus present from the successful reads for the flat-coated retriever. And for the husky, 38.8% remained unclassified at the kingdom level, but we were able to still get 61.2 characterized as bacteria. And so you can see for the husky, a large portion were unclassified at the kingdom level, but when we look at the results from the order, you can see the top order classification for the flat-coated retriever up here. And so we have a pretty large mix, the most being Bacteriodales, which makes up 43.5%. We have Fusobacterials, which is 22.09%. The Clostridales, I hope I'm saying these right, at 16.94%. We had 7.9% unclassified, 3.93% as the Flavobacterials, and then other was 5.6%. And the other category on this means that it was made up of bacteria that was smaller than, I believe, 3.5%, so it gets grouped into the other classification. So for the husky, we have pretty much the same. You can see here that comparing by order, both dogs have the Clostridales, the Flavobacterials, the Bacteriodales, and a portion of the unclassified bacteria living within their gut microbiomes. And the main difference is that the flat-coated retriever has an order called Fusobacteriodales, which could be a potential sign of disease because in humans, Fusobacterium should be treated as a pathogen if found in the gut, and it is also associated with ulcers in the colon. And so, as I said, the other categories on these charts are the sum of all the classifications with less than a 3.5% abundance in the microbiome. And so on a genus level, you can see here that we have these bar charts that show the different genus classifications for the samples found in the gut microbiome. And the main, for the flat-coated retriever, it has the largest amount as Bacteriodales. The second largest amount is actually unclassified at the genus level. And then from there on, it decreases with Fusobacterium being present pre-Votella, Cetobacterium, this is a fun one to say, Fasciculobacterium, Faecilibacterium, and Flavobacterium being the smallest amount. And so last but not least, for the genus classification for the husky, the largest percentage that we had was actually unclassified at the genus level. And the next biggest was the Plautia, and then after that was the Flavobacterium, which was in common with the flat-coated retriever. And so our DNA extraction in total for this was successful in terms of quantity, and we could tell this based on the absorbance reading when we measured with the UV spectrophotometry. And as I stated earlier, we used the 260 to 280 nanometer ratio to tell, and all of our samples were considered pure between 1.5 and 2.2. And so the results of our Amplicon PCR were shown through our gel electrophoresis, and we were able to tell that our Amplicon PCR was successful because all the bands were brightly colored except for the negative control. And they were all at 550 base pairs, which is exactly what we expected. And the index PCR was used to add individual tags to our samples. And to do this, we needed accurate concentrations of our DNA in our samples. And I have previously stated those with the chart that I have available. And though we were unable to sequence our own, we were able to analyze the gut microbiome and compare the husky to the flat-coated retriever.